Welcome back. We're up to Lesson 5, What is RNA? In this lesson, we'll make models of messenger RNA, and this video should go a lot faster than the other one. Please do all your background reading, and in your background reading, you'll see a delightful metaphor of building a house. And your DNA is like your blueprint that's locked tightly into the safe in the architect's office. Your messenger RNA copies and brings that DNA out to the world, and your transfer RNA and your ribosomal RNA begin building proteins, which comes into the next lesson. So you've got a lot of background information to read, and in that background information, you will see some important differences in DNA and in RNA. And there is a wonderful video that you'll want to show your students. One of my favorite parts of this video is showing these bits of floating nucleotides. That kind of helps bring home the idea of the nucleotide soup. This enzyme would be helicase, that is unzipping the DNA as it's transcribed, and then notice the DNA rezips as it leaves that molecule. Great stuff. Okay, so here these slides will take you through your differences in DNA and RNA. And notice in your DNA molecule, you're missing an oxygen, deoxy. And your connects blocks beautifully show an extra little bit on your ribose sugar group. Here you've got a nodule or a node that can represent that piece of oxygen that is not present on your DNA. Isn't that kind of nice when models match up that well? And of course, you're going to have a different nitrogen base. You'll have uracil instead of thymine. And as with your previous lesson, please assemble the nucleotides before making your messenger RNA. So following the same rules of base pairing, except of course you're using uracil, you will build a model based on your original instructions. Students are still working in the same groups with the same worksheet. You will use this strand. This complementary strand is what the RNA will use to transcribe from. So again, you'll build from 5' to 3', and do this on paper first. Now, students may notice as they match up base pairs one at a time that their messenger RNA sequence matches their original DNA sequence, except for they have U's where there were T's. And that's fine if students notice that. And that is the end result of this transcription, but you don't want to tell students just to take that shortcut, because that is not the process that goes on in the body. Of course, the process that goes on in the body is these two are unzipped, this strand is transcribed, and then these two are rezipped. So yes, you do end up with an almost exact copy of this DNA, but again, the process is different. So there's some terminology for you. You'll want to build your nucleotides just like you did ahead of time with your DNA building. So again, you might want to tell a little story of your hand going along and unzipping these two strands of DNA with your helicase hand enzyme and building, starting from the 5' end, one nucleotide at a time. And once your strand of messenger RNA is built, you can separate this whole strand, out it goes, and the DNA begins to zip back up together over here. And that's pretty close to you're going to be able to do, about as close as you're going to be able to do with this model. You'll put your DNA models back on your stands. There's some background information, and of course, you always want to be moving toward your course of study standards. This concludes Lesson 5, and we'll see you back to make some protein in Lesson 6. Thanks for watching.